Picking up where we left off in Chapter 5, we're looking at the selection of different antimicrobial procedures, keeping in mind that most of these are specific to bacteria and viruses. The ideal method does not exist. That goes back to the idea of a standard operating procedure and somebody that is in charge of all the different types of sanitation and antimicrobial procedures available, because each one has its drawbacks and procedural parameters. Choice depends on a lot of different factors, as we talked about last time. What kind of microbes are we expecting? How many different kinds are we expecting on top of overall how many? Environmental conditions, in terms of, are we dealing with outside, are we dealing with inside, are we dealing with humidity, which one of these or any of these is going to be applicable. Risk of infection, this goes back to the idea of a BSL 1 through 4 agents and how dangerous is the situation that we're getting ourselves into. And composition of the infected material. Is it made of wood, plastics, metals? Each of these are going to have their drawbacks based on what procedures are available. Most labs have somebody involved with that, as we just talked about. Next slides, we're going to look at some of the things that we consider. So types of organisms. This listing here is the ones that are incredibly difficult to clean with standard procedures. Therefore, we have to come up with some alternative methods if we're expecting any of these to be available. So endospores we talked about in the past, mainly the Bacillus and the Clostridium genii. Only extreme heat or chemicals completely destroy them. So this is going to involve the autoclave typically to try to maximize our chances of eradicating all endospores. Protozoan cyst and oocyst, well, these are the names of endospores that are given to eukaryotes, specifically to protozoa. We call them cysts instead of endospores. So same idea, very resistant to disinfectants. This is how they're able to pass on from person to person, usually in fecal-oral route, excreted in the feces. Diarrheal diseases are typically involved with protozoa. Mycobacterium, remember these are the ones that actually have the mycolic acid as their composition of the wall outside of the phospholipid bilayer, instead of it being made of peptidoglycan like the gram-negative and gram-positives. So Mycobacterium, that mycolic acid is waxy, hydrophobic, makes it a lot more difficult for cleaning chemical treatments. Pseudomonas aeruginosa specifically is one of the biggest hospital-acquired infections that we'll see as we move forward, especially when we get into respiratory and wound infections. Pseudomonas can actually grow in chlorine, and so it makes it very difficult to clean to ensure that it's all eradicated. So a lot of procedures involve minimizing anything that would potentially bring Pseudomonas into the hospital settings. If grandma's in the intensive care unit, you're not going to be able to bring flowers or anything else from the outside because Pseudomonas could possibly be on it. And naked viruses, these are ones that are going to lack an envelope, as you'll learn about in Chapter 13. These are much more difficult to also clean. So how do we actually determine which antimicrobial procedure to pick? Well, there's what we call the D value. This is the time for heat and chemicals to kill an affected population. What I want you to notice is that it's actually in logarithmic scale. So what we're looking at is every time that we go over or go down, I should say, on the y-axis from, say, 10 to the 8th to 10 to the 7th, that's a log decrease. And so that's actually telling us that is almost 90% of the population has been eradicated each time we go down a exponent. So larger the population, so that's what you're seeing there on the y-axis, is how large is the population. And so the larger the population, the more time it's going to take, right? We have to clean them all out. So that's what we call decimal reduction time. Time required to kill 90% of the population under specific conditions. So as you can see, they're going from 10 to the 8th to 10 to the 7th. Gives us a D value of roughly 15 minutes. Going from 10 to the 3rd down to 10 to the 2nd is also 15 minutes. So the D value for this particular chemical is 15 minutes that the chemical would actually have to sit on the perceived contaminated surfaces. So environmental conditions on top of it being inside-outside type ideas. It could also be dirt, grease, if we're looking at something that might be like a mechanical area, body fluids, if we're looking at hospital clinical areas. Any of those could potentially disrupt the ability of heat to actually do its job. So it's important to thoroughly clean those outside areas before we actually go and start thinking about ensuring that the surface is completely clean or sterilized. So one of the things that we like to point out is turkey processing facilities. If you were to go to one, there's one up in Laurel, Mississippi. You go to these facilities, and what they do is they work all day prepping the turkeys, but at the end of the day, a cleaning crew comes in and dismantles the entire production facility down to the individual screws, cleans them, and puts it all back together again. So it's ready to process more turkeys the next day. One of the reasons we do that is salmonella. Salmonella is ubiquitous to poultry, and so just a little bit of contaminated meat, if it was stuck, like say, in a roller of the conveyor belts, that little piece of meat would potentially touch every single piece of meat going by and contaminate it. So it's incredibly important that the cleaning crews do a very thorough job every day. pH and temperature can also influence effectiveness. We mop usually with warm to hot water. Why? Because these solutions actually can increase the speed of killing the microbes when we add that heat in on top of the actual cleaning agent. So here's sodium hypochlorite, which is just a fancy name for bleach. We can kill a suspension of tuberculosis at 55 degrees Celsius in half the time that it takes at 50. So just those 5 degrees Celsius, which is roughly about 12 degrees Fahrenheit for those of you that aren't quite up to speed on your Celsius. Just that little kick up in temperature is enough to half the amount of time that the bleach actually has to sit on the surface. So risk of infection. This isn't necessarily for the cleaning crew as much as it is for, say, a clinical situation. We're dealing with patients, and so here we have three major criteria. Critical items. These are ones that are going to come into contact with body tissue. Typically, body tissue is sterile, so anything that goes in and touches body tissue has to be sterile also. So this is going to include needles and scalpels. Traditionally nowadays, they're one-time use utensils, so you're not going to have to necessarily worry about cleaning them. It's much more just easier, more sanitary to just throw them away after one use. Semi-critical instruments. These are ones going to contact things like mucous membranes and whatnot, but do not necessarily, actually break the skin to go into the tissue level. However, you still are inserting it in things. So this would be like probes. And so for these, we have to make sure that the viruses and vegetative bacteria are cleaned off of them. When we use the word vegetative, that means ones that are actively growing versus ones that are in an endospore. So endoscopes and tracheal tubes, these would be what we call semi-critical. Non-critical instruments are going to be ones that come in contact with unbroken skin only. So this would be like a stethoscope, blood pressure cuffs, countertops, very low risk of transmission for these. Composition, as we talked about before, composition of an item is going to play a big role in which cleaning and antimicrobial procedure we can use. So heat, of course, would be inappropriate for plastics. Surprisingly, a lot of our laboratory equipment is made of plastic. And so it takes a whole different level to clean. We can't use heat with that because, of course, it would melt. Irradiation is good. Using UV light traditionally is what we use. However, some plastics over time become brittle when exposed to UV. And moist heat, this would be using things like an autoclave, cannot, of course, be used if we're dealing with moisture-sensitive material, like, say, media that we're growing bacteria in. We can't necessarily use moist heat unless we do some more procedures prior to putting it in the autoclave. So heat treatment's useful for microbial control. It's safe, fast, and inexpensive. We can use just what looks like almost like a toaster oven that we use in the laboratories that we put our metal tools in at the end of the day. Can be used to sterilize or disinfect. That's just going to come down to the amount of time that you actually have it in the heat. And we can use moist heat or dry heat. Dry heat would be using the oven systems like I was just describing. Moist heat would be using things like an autoclave that we'll see coming up. So, benefits of using moist heat. Moist heat is going to irreversibly denature proteins. That's going to include toxins. That's going to include a lot of other potentially dangerous proteins that the bacteria could be making that could lead to sickness. And so having them irreversibly denatured ensures that those proteins are not going to just accidentally or on purpose reform and potentially be back to their same destructive protein. And so boiling destroys most of the microorganisms and viruses. However, it does not get rid of the endospores. Pasteurization, which we talked about briefly, destroys pathogens and the spoilage organisms. But if we were to use it for too long, it could potentially change the flavors and texture of the foods, which then is going to be a marketing issue. So we have two different styles of pasteurization. We have high temp, short time. This is what most things that we're used to buying from the store that say they're pasteurized. That's what they've been put through. So this is going to be things like milk, 72 degrees Celsius. Remind yourself, Celsius scale, boiling is 100 degrees Celsius. And so 72 is going to be roughly about 180 degrees or so for 15 seconds. Ice cream a little bit hotter for about 20 seconds. Of course, this is going to be before it's frozen into its ice cream. This is when it's just its cream when it's being produced. Now, the other one that you see there is ultra high temp. This is going to be for boxed juices. So any of your juices that you buy at the store that are in like Capri Suns, things like this, are going to be typically done with a ultra high temperature. And so this is going to be also milk. So like when I've worked overseas, a lot of your milk is actually bought on the store shelves versus in a cooler. Biggest problem overseas, especially in third world countries, is going to be cold chain. And so not having the ability to actually store things cold from the actual point of production to the store has led to other ways to take care of these food products. So milk can be ultra high temp for 140 degrees Celsius, so way over boiling for a few seconds and rapidly cooled. Now, once you get over that boiling level, it becomes problematic because you're going to be changing the flavor. So these milks don't taste anything like milks that we're used to tasting. All right, so autoclaving is going to be sterilizing using pressurized steam. And so this is going to use both pressure and temperature to ultimately destroy or sterilize anything you put in it. And this is the way that we can get rid of endospores. And so here, the traditional sterilization time and temperature is going to be 121 degrees Celsius at 15 psi. Psi stands for pressure per square inch, and so standard pressure is 1. So this is 15 times the amount of pressure you feel on yourself right now that these instruments, whatever you put in here, will be subjected to for about 15 minutes. Now, it takes time for it to come up to pressure and time for it to release that pressure, so the overall time that it's actually in the autoclave is going to be much longer than 15 minutes, but the amount of time it's at pressure actually doing its thing, sterilizing, is going to be for 15 minutes. Prions are thought to be destroyed at 132 degrees Celsius for about an hour in the autoclave. The actual jury is still out on how many could potentially still survive even at those temperatures. All right, so moving up to actual commercial canning, we have to remind ourselves that food canning has really only been around since the 1950s. And so what they would actually use back when canning was first started would be pressure cookers at home and mason jars, and essentially saving any extra gardening foods, vegetables that you made in the garden that can be then canned and saved over the winter time. However, using mason jars at the industrial level is going to be a no-go. And so what they have come up with in terms of the cans that we now go buy at the store is using what's called a retort. And so retorts are very large rooms almost that can do multiple cans all at one time. And so the idea behind these is to destroy the Clostridium botulinum. Remember, Clostridium is one of them that can make endospores. That's why we're focusing on this one. We want to reduce the potential of having 10 to the 12th. That means 10 with 12 zeros at the end, so 1 trillion endospores to only 1, and that's why it's called a 12D process. Remember, the D value is the amount of time it takes to go down just one logarithmic scale We're seeing how long it takes to go down 12, and specifically looking at the Clostridiums. And so it's virtually impossible, one, to have that many endospores. So going 10 to the 12th and having everything based off of that number is going well and above the call of duty when it comes to what's actually necessary to clear. We're not going to see anything to those levels. Maybe 10 to the 8th might be the highest we might see. Critical because otherwise endospores can germinate. So one of the biggest problems when commercial canning first came around, and you may have heard these stories, you don't buy dented cans. Well, nowadays a dented can just means that somebody probably dropped it and put a dent in it. But back in the 60s and 70s and even into the 80s, having a dented can suggested that you might have botulinum in the can. Clostridium is what we call an obligate anaerobe. And so it prefers actually growing, and actually obligate meaning that it has to grow in these non-oxygen environments. So being in a sealed can is one of the best situations that Clostridium can have. Why do we worry about Clostridium botulinum? Because one, it is found in soil. That's where it's actually typically found. And so where do vegetables come from? They're going to be growing out of soil. So the potential of having Clostridium contamination is pretty high when you're dealing with vegetables. Two, it produces a toxin, and this toxin is incredibly deadly. And so by looking and concentrating all our efforts just on this one Clostridium species, pretty much ensures that the rest of the species of bacteria that we might be concerned with are going to be gone also. Dry heat, this is just using an oven. As I said, sometimes we'll actually leave things in much longer than what it suggests here. 200 degrees Celsius for 90 minutes or even longer. Sometimes we'd even have it a little bit cooler and leave it overnight. And we're going to use mainly just metals for this. What is it going to do? It's also going to burn things up. So if we wanted to go higher, we can use incineration. And so this would be using live fire. So we can use live fire to oxidize any leftover bacteria that might be on an inoculating loop. By burning it up, you ensure that there's nothing left alive. You take it down to ashes. Biohazard kits, biohazard waste containers traditionally are going to be lost in an incinerator device that you may have on the actual campus. Now we can also use filtration, and this is going to be used mainly for medias, things that we're going to be growing the bacteria in. One, we don't want to destroy any of the proteins or any of the growth media that might be in there. And so filters are going to be the best option. The smaller the filter, the pore size on the filter, the better off. What we traditionally use is what's called a 0.2 micrometer filter pore size. And what that is going to minimize is any chance of any bacteria getting through. Most viruses are not going to be able to get through there, but there's a chance you might. And so depending on what you're actually trying to filter out, you may go smaller, but traditional go-to filters are the 0.2 micrometer membrane filters. Now we can also do depth filters, and that's going to be going through a very thick filter where you're going to have multiple porous materials in there that could potentially catch or even more. And also you might even have electrically charged. And so if you're looking to get rid of cells, remember the majority of the phospholipid bilayer is negatively charged. And so having positively charged filters would almost ensure also to pull away any living organisms that may be going through your media. The last one down there, filtration of air. A lot of people have heard of HEPA filters. HEPA just stands for high efficiency particulate air, trying to remove all the microbes from air. Going back to chapter 1 in Louis Pasteur's work, discovering that these microbes actually travel on dust. Radiation, as I said, there's lots and lots of different types of radiation, but what we're looking at here is going to be UV light. UV light is going to be the one that is used. The blue lights that you may be familiar with if you've ever seen UV lights before. Putting anything in there into like a UV chamber, turning on the light, what it's going to do is it's going to destroy the DNA of any organism that's hit with that UV light. What happens is, remember, you got 4 nucleotides, guanine, thymine, adenine, and cytosine. Anytime you have two thiamines next to each other that's hit with a UV light, they'll actually cause them to bind to themselves versus binding to the alternate strand, to the adenine that's on the other side. Remember, T's and A's and G's and C's bond to each other. The thiamines that are next to each other will bond and make what's called a thiamine dimer, which then causes the integrity of the DNA to start to be compromised. Enough of that happening along the strand of DNA essentially makes the DNA useless, and the organism ultimately dies. Some other ways that we can use it is ionizing radiation, and so this is going to remove the electrons from atoms by using a positive charge, and this is going to also destroy DNA, destroy the cytoplasmic membrane, and also is going to cause the formation of reactive oxidative species. Remember, we talked about this in a previous class, is that water and oxygen are two of the most common molecules found in a living organism, H2O and O2. Reactive oxidative species are going to be things like hydrogen peroxide, H2O2, superoxide, H2O3, and ozone, O3. All three of these are unstable molecules, and they will produce these reactive oxidative species. They get their name reactive because they're unstable. They're looking for a partner. They want to go back to being just water or oxygen. And therefore, that being reactive makes them destructive and can also be beneficial to removing microbes, but too many of them can be dangerous to the actual person too. So gamma rays and X-rays are important also. We use to sterilize heat-sensitive materials. Generally after packing, some of these things are going to be used on foods. However, customer pushback has limited their use. A lot of people start getting freaked out if you put too much radiation on their food. FDA has mainly approved it for parasite control and bacterial control in meats. But after that, there isn't much use for radiation. We already talked about ultraviolet. Microwaves kill by generating heat, not directly. Just like heating up your food over time. If it's a very large product that you're putting into the microwave, it's going to heat unevenly, which then potentially could allow bacteria to still survive. So microwaves are not the most ideal situation for sterilizing. High pressure, this is going to be the same thing we talked about before, but now we're going even higher. And so this is going to be limited to things like hummus, guacamole, things that are creamy, because you're using so much pressure in this situation that anything else is going to be turned into mush anyway. So employs high pressure up to 130 psi. What are we doing here? Well, we're minimizing the need for heat, because heat would basically ruin guacamole, would ruin hummus. But if we increase the pressure from 15 psi that we find traditionally in an autoclave up to 130,000 psi, then we're going to ensure that there's going to be nothing living there either, because 130,000 times the amount of pressure that you feel right now is going to crush anything. And so ideal for those type of foods. Anything else would just be turned into mush and would not be a good use of high pressure. So here's a table. It has all the different ones that we talked about. A good place to start for any sort of study purposes. All right, so using chemicals to destroy microorganisms and viruses. Here we're just going to go through a small laundry list of different chemicals, but they're all going to fit into these four major categories. They're either going to be sterilants, which destroy all microorganisms. Going to be very useful for heat-sensitive critical equipment. Remember, critical instruments are ones that are going to be actually touching body tissue. And so we'd want to use something that's going to destroy all microorganisms. High-level disinfectants, destroying viruses and vegetative cells. Remember, vegetative means actively growing, not in an endospore. This is going to be useful for semi-critical instruments such as the trach tubes. Intermediate level disinfectants, destroying vegetative, mycobacteria, fungi, and most viruses. Useful for non-critical also, using it for countertops, using it for endospores. It's going to be mainly like wipes and stuff that we use. And then low-level disinfectants, this is going to be things that we use for furniture and floors. People that come in and mop the floors, and maybe putting down some carpet cleaner, perhaps if it's a carpeted room. Furniture wipe downs. This is going to be a low-level disinfectants. So what do we have to assess? Well, this is why somebody that is an environmental scientist working in, like, say, a hospital setting doing those standard operating procedures, they get paid pretty well, because there's a lot that you have to consider for each chemical that you're using. One, toxicity. Benefits have to outweigh the risk of use. Yeah, sure, it may be the greatest sterilant in the world, but touching it may be toxic and cause you to have health problems. That's not a good chemical to use then, and so that would be one that would be probably kicked out. Activity. How active is it in terms of presence of other organic material? So is it going to sit there and let off fumes? Is it going to react with anything it touches? Is it going to melt rubbers but clean carpets, clean tile really well? Carpet's a bad example. Compatibility. Are we mixing chemicals with things that can't necessarily get wet? So like electrical equipment, you have to use different types of cleansers when dealing with electronics versus a countertop. Residues, does it leave anything behind? Is it leaving some sort of film behind that could potentially be toxic or corrosive if it's on metal? Cost. Well, just because it's 10% better than the next possible sterilant out there, but if it costs 20 times as much, then the cost is going to outweigh the benefit, and so these things have to be put into play just like anything else in our life. Storage, well, different types of chemicals require different types of storage. They may not be able to be stored in the same spot, and so you don't want to have 15 different chemicals that all have very specific storage requirements, which then is going to become costly to the employer, in terms of storage. So we want to make sure that we're getting things that can be stored together. Environmental risk, is there anything that we have to worry about in terms of, can it go down the sink? If it can't go down the sink, where does it need to go? What is the actual process of disposing? Do we have to contract with a company that comes and takes care of it, or is it something that we can do on our own? For instance, putting it in the autoclave before dumping it down the sink, which is some of the things that we might have done back in the microbiology lab. So some of these different chemicals, alcohols are pretty ubiquitous, in laboratories. It says 60-80%. I'm going to tell you right now, the most ideal percent of alcohol is always 70%. It can either be ethyl alcohol or it could be isopropanol alcohol. You can typically buy either one of those at Walgreens for either at 90 or 100%. So you just dilute it down with standard water till you get to that 70%. And that is ideal. Why? Because it doesn't evaporate very quickly, and it is still as high as we can get to be effective at getting rid of the bacteria or other living organisms that might be on what we're cleansing. And so we would use this mainly as a surface cleaner. So laboratory surface, we used over in the freshman labs and microbiology labs. We wipe them down with ethyl alcohol before we get started. Aldehydes, this is going to be like formaldehydes, glutaraldehydes. Biggest thing that these guys do is they freeze proteins. And so a lot of the proteins that are necessary for biological organisms is to have these proteins be able to contort a little bit like an enzyme, and so being frozen in place and activating them is going to essentially kill the microbe. And so here, the most common is about 2% solution immersed for about 10 to 12 hours. We can also use gas formalin at 37% solution. This is going to be used to preserve specimens. So anything that's being preserved is usually going to be preserved in formaldehyde, fixing those proteins, preventing it from decomposing as fast as it would naturally. Bioguanides, these are going to be things like chlorhexidine. It's going to be the most effective. It's very, very common in antiseptics. So if you went and looked in your first aid kits, if you have any antiseptics in there, useful for like cleaning skin, that might be you might have some sort of rash or something on it. This is going to be the type of antiseptics that you're going to use to destroy vegetative bacteria, funguses, and viruses. Also found in mouthwashes and things like this to try to knock down infectious disease agents. Ethylene oxide, this is a gaseous sterilant that destroys pretty much everything. This is going to be very useful for pacemakers, artificial hips, anything that's being inserted into a person for the duration of their life, or as long as we can get it to stay. This is what we're going to clean with, but of course it's going to be very difficult to work with because it is mutagenic and potentially carcinogenic. And so these are going to be things that happen before they typically leave the facility that they're produced in. So if I went in order to sleeve of Petri dishes, those Petri dishes will be hit with ethylene oxide before they're actually packaged in their plastics and then sealed in their plastics. So when they get to me, if there was not any sort of major breach in the packaging material, then they should still hold their the effects of the sterilization using ethylene oxide. Otherwise, this is not something that would typically be used by any just normal technical group or laboratory group. Halogens, these are going to be things like chlorine and iodine. They're going to be used as a disinfectant, usually at a 1 to 100 ratio. And this is going to also be used even at very low levels in our drinking water. So one of the last things that the water is hit with is a little bit of chlorine solution to try to just knock out any potential microorganisms that might still be there. However, as we talked about with the sewage and water treatment plants, having those byproducts formed is a potential negative for using chlorine. Iodine used to be very common, nowadays still not used as readily. So both of these would be used as secondary, not typically just a primary cleaning agent. It's going to be used in conjunction with other things. Because as you can see there, Pseudomonas, Cryptosporidium, and Gerardia can all survive in either iodine or chlorine, so it makes it pretty unreliable to use as a primary cleaning agent. Metal compounds, this is going to be things that have typically copper in them, but it can also be mercury and tin. What happens is the bacteria cannot survive. This is one of the reasons why we have copper being used in our refrigeration, whether it be AC or refrigerators. Copper is a natural antimicrobial agent, and so having those metals, and having water running through them or refrigerant and running through them keeps them sterile as best as possible. However, high concentrations of these metals can be too toxic to be used medically, and so we have to use them in very limited amounts. One of the more common ones that is used on a regular basis is silver as a disinfectant, used in a lot of creams and bandages as a silver nitrate. This is going to be something we also use in eye drops to children that are born. Immediately they're hit with these 2 eye drops. We'll talk about when we get up to those chapters. Those eye drops are there just in case the mother might have a gonorrheal infection or other STD infections that prevents it from getting into eyes and ultimately blinding the child for life potentially. Using these eye drops is a good way to try to minimize any of that vertical transfer from mother to child. Ozone, this was pretty popular for a while. People would have home air filtration units that had an ozone feature on it. Again, ozone is a very unstable form of oxygen. You can see there's O3. That means there's an extra oxygen. Oxygen likes to be as O2. And so this is a powerful oxidizing agent because that extra oxygen is going to bounce off and cause breakdown of anything that it comes in contact with. So specifically like a phospholipid bilayer cell membrane, potentially rip it apart of a bacteria, and all you need is just one little breach in the phospholipid bilayer to ultimately kill the bacteria. Next one here is paraoxygens. This is going to fall underneath the reactive oxidative species also, mainly looking at hydrogen peroxide. It used to be considered one of the top chemicals you should be having in your first aid kit. However, a few years ago, the CDC came out and said, don't use it as much as we used to be using it, because not only does it destroy the infectious material that might be in a wound, but it's also damaging the healthy tissue because it's just a powerful oxidizer. Again, hydrogen peroxide wants to break back down into oxygen and water. It doesn't want to stay as a hydrogen peroxide. Another one, peracetic acid, is a lot more potent than hydrogen peroxide, therefore not easily available to the public. Typically going to be something you want to purchase through a vendor. Phenolic compounds, these are going to be phenols. This is one of the very earliest disinfectants, as we've seen there at the beginning of the chapter with Lister coming up with realizing that patients potentially needed to be cleaned, or materials need to be cleaned before they're used on the patient. One of the very first ones he used was phenol. Has a very nasty odor and irritates the skin if it gets on your skin. And so it's not going to be great, as it's not going to be used as much as it used to be. However, it's very good at killing vegetative bacteria. It can kill mycobacterium in high volume concentrations. However, it's not reliable at all with different virus groups because it's going to destroy the cytoplasmic membrane. Viruses, again, do not have a cytoplasmic membrane. It has a lot of different activity, reasonable cost, remains effective in the presence of detergents and other organic contaminants. So using it by itself is not really ideal, but used in combination can be potentially useful still. Some of the ways it's also used is in a non-toxic form, used in different soaps and lotions, given the name triclosan or hexachlorophene. Quaternary ammonium compounds. These are cationic, so these are going to be positively charged detergents. So the same idea as going after the cytoplasmic membrane. So this is non-toxic, can be used to disinfect food preparation services. This is going to be the same as some of your cleaning agents you may have underneath your kitchen counters. Reduces the surface tension of different liquids, helps in removal of dirts and organic materials. Most household soaps and detergents are going to be anionic, so that means they have more of a negative charge. However, the positive charge of quaternaries are going to attract them to the negative charge of the cell surface, which is very good. Soaps and detergents being anionic, relying more on their hydrophobicity to break down the cell membrane. So here's your class of all your different chemicals. Again, these tables are a good spot to just test your knowledge, understanding that, again, I'm not going to ask you questions that are going to be very specific. So knowing the pros and cons to each of the chemicals is going to be the extent of what I would be asking you any questions on the exam. All right, so preservation of perishable products. So it's going to be mainly fruits and vegetables, meat products. Chemical preservatives are used, but most people don't like that. They're going, when they hear there's chemical preservatives in their food, a lot of people shy away, and that's why you got these new organic produce sections, meat sections, things like this, minimizing any sort of chemicals that may be used in the food. So some of the things that we can use: weak organic acids, so benzoics, sorbics, and propionics. This is going to inhibit metabolism, so it's going to slow down the growth. Again, it's not going to kill, it's just going to slow down the speed that they're able to grow, and so this could be not only infectious agents, but also ones that are involved with spoilage. So nitrate and nitrates used in processing meats inhibits endospore germination and vegetative cell growth. So it's going to stop the growth of Clostridium botulinum, and it also gives meat its pinkish color. However, over time, we have shown that these nitrates can potentially be carcinogenic, and so a lot of products are shying away from nitrates. So places that we used to see them quite a bit: hot dogs, any sort of sausages, things like this, processed and then packaged. Usually if you look on it, there'll be some sort of nitrate that's used as preservative. However, again, moving away from those because of the potential of them being carcinogenic. Low temperature storage is what we all do. There's nothing secret about this, just using refrigeration. What does refrigeration do? Slows down the growth of the bacteria. Bacteria like to grow at a certain temperature. You cool them down to refrigerator temp, now they're going to go a little bit slower. It's not going to kill them. So it's going to just keep them from growing at the speed that they were growing. However, there are some bacteria that are termed psychotrophs. Psychro just means that they like growing in cold temperatures. Thermo would be the opposite for ones that grow in high temperatures. And meso, M-E-S-O, is the term that we give to bacteria that like to grow at our temperature, so middle. Again, these are all based off of human. So do they recognize it as cold? No. Do they recognize it as hot? No, it's their temperature that they prefer. But for us, basing everything off of the human condition, we use the terms psychro, meso, and thermo. Reducing available water, this would be things like doing dehydration to make jerkies, making freeze-dried fruits and vegetables. What we're going to do, this is going to reduce the available water. The water not being available hinders the ability of the microbe to grow. And so we can accomplish this by adding salts, sugars, drying the food out, as I was talking about. And by doing that, it's going to cause, by adding the salts and sugars, is going to cause the cells to become hypertonic. All their water gets removed and ultimately will kill the cells. And so this is a very safe way to store meats, especially. However, some bacteria, as I've been pointing out the entire first 2 weeks of class here, there's always an exception to every rule. And so in this case, the exemption is going to be Staphylococcus aureus. They prefer growing in salt conditions. This is going to be, as we get up to the skin chapters, we're going to learn that the skin has quite a bit of Staphylococcus on it. It has Staph aureus as well as epidermis, and they're there as part of the natural microbiota, fighting off any other sort of bacterium that could potentially try to establish residency on the skin. The staph species likes higher salt concentrations. Our skin has a natural salt concentration to it. Lyophilization is the fancy term for freeze drying, and so you'll see that down there on the 5th bullet. And so it's just a term to keep yourself familiar with. So you go buy a can of or a package of coffee that's already been ground up. Well, the coffee has actually also been lyophilized to pull out any sort of water to keep it from spoiling. We can also get milk that has been powdered milk, fruits and vegetables that have been dehydrated or lyophilized for storage. Drying stops microbial growth but does not reliably kill it, so we have to keep that in mind also, that for instance, some people actually like to use freeze-dried eggs, and if the salmonella was already infected on the egg, because it's common to poultry, drying it out is not going to reliably get rid of all the salmonella, so sometimes some secondaries need to be taken care of in order to make the food safe. So contamination of an operating room, you're looking here, there's lots of different things happening in that picture. You got not only the floors, you got the surface where the instruments are going to be kept, you got the actual gurney for the patient, you got the storage area over there in the back. All those are going to require different cleaning agents as well as different procedures, again going back to having a good standard operating procedure for your place of employment. All right, so that concludes Chapter 5, so that's going to give you all the information you need for Exam 1, which is going to include Chapters 1, 3, and 5. Good luck, and if you have any questions, feel free to email me.